Hi, I'm Dr. Fox, and let's talk about nephrons and renal histology. In this video, we'll discuss the nephron, its parts, and how it functions. We'll also discuss a little bit about how substances are either secreted or reabsorbed by the nephron. And finally, we'll discuss how diuretics affect urine formation. Let's take a look at the nephron. The nephron is the functional unit of the kidney, and there are millions of nephrons in each kidney. Now, nephrons we can break down into three distinct parts. There's the renal corpuscle, the tubules, and then finally the collecting ducts into which the nephrons flow. So the renal corpuscle has two parts. There's a glomerular capsule, and then there's the glomerulus, which is a specialized capillary bed. So afferent arterioles are coming from interlobular or cortical radiate arteries feeding into the glomerulus. So it's here that filtrate is formed. So this is a process known as filtrate formation or filtration. So blood gets converted into filtrate. That filtrate then moves through a series of tubes. And as it does, there are many instances of two distinct processes which will occur: tubular secretion and tubular reabsorption. With tubular secretion, we are going to take the opportunity to push more substances into the filtrate. So we have materials that are in the blood, in those peritubular capillaries, being secreted into the tubules to join the filtrate. Then we have tubular reabsorption. And with tubular reabsorption, we make a decision that we don't want to lose the substances that are in the filtrate. And so we're going to rescue portions of them and move them back into the cardiovascular system. We'll then move the filtrate through the collecting duct, and at the renal papilla, that filtrate then becomes urine. So we have filtration, tubular secretion, and tubular reabsorption. The formation of filtrate, then with secretion, we have adding to the filtrate, and with tubular reabsorption, we have rescuing things from the filtrate and putting them back into the blood. So there is this dance between what is filtrate and what is in the blood. Let's start with the renal corpuscle. So we've got the capsule, and then we have the glomerulus. Now, between the glomerulus and the capsule, we have a serous membrane. So there is a visceral layer and a parietal layer. The visceral layer is adhered to the glomerulus. The parietal layer is adhered to the capsule. So the afferent arteriole moves blood in. That blood is transformed into filtrate and moved along the tubules, and the efferent arteriole moves blood out towards the peritubular capillaries. The rate at which blood is transformed into filtrate is called the glomerular filtration rate. We can shorten that to GFR. And GFR is going to be proportional to blood pressure. Now, the filtration membrane that's responsible for glomerular filtration is complements of two distinct parts. So first, there are podocytes. Podocytes have little limbs. That's why they're called limbed cells, or podocytes, that sit upon the glomerulus. So this is that visceral layer of the membrane there. And their little extensions, or their legs, as we can see here, are what forms that filter in helping to keep all of the formed elements in, whereas letting other substances, smaller substances, through. There's also specialized smooth muscle in and amongst these called mesangial cells, or mesangium, that can bring them closer together or relax and have them further apart to help fine-tune that filtration barrier, and thus help to either increase or decrease glomerular filtration rate. So they will relax to increase glomerular filtration rate, or they will contract to help decrease glomerular filtration rate. The glomerulus is going to push filtrate into, first, the proximal convoluted tubule. So the PCT for short. There's two parts to it, a convoluted part and a straight part. So this is the pars convoluta for the convoluted part and the pars recta for the straight part. And so as you notice, as we go through here, different processes are going to occur. So the proximal convoluted tubule is the principal site of tubular reabsorption. So that is pushing materials from the filtrate back into the blood. So we are going to reabsorb most of salts and most water here. We'll reabsorb 100% of glucose and also amino acids here, 80% of citrate, 80% of phosphate, 65% of potassium, and we'll reabsorb about half the urea here. We'll also have processes of tubular secretion, so any bioactive substances such as medication or ammonia-rich substances will be pushed from the blood and into the filtrate here. This proximal convoluted tubule is then going to move into the nephron loop. The nephron loop, or the loop of Henle, is a place where we can further refine and reabsorb water. So here's the loop as we go, and that loop dives deep into the medulla of the kidney. And what happens is, as we move filtrate along through this loop, we're going to move water out into the surrounding spaces. Now, we move water out because we make this tissue hyperosmotic, and we do so by increasing the concentration of sodium ions. So sodium ions are first going to passively exit the loop, and then we'll actually use ATP to actively pump sodium out. And by making this an increasingly higher concentration gradient, we pull more and more water out from the filtrate. So we've got a lot of tubular reabsorption of water going on here, and that water gets picked up by the vasa recta, which then delivers it back through venous return. So desert-dwelling organisms typically have more nephrons with more distinct loops of Henle within them. The nephron loop then feeds into the distal convoluted tubule. The distal convoluted tubule is going to head towards the collecting duct, and we have many opportunities for either tubular secretion or tubular reabsorption. And oftentimes these processes are dependent upon specific types of cells. So there are two main types of cells that are important in the distal convoluted tubule: intercalated cells and principal cells. Intercalated cells are going to be important for the maintenance of pH homeostasis. So they're capable of either secreting protons or conserving bicarbonate ions, so moving them between the blood and the filtrate depending on what conditions require. And then principal cells are going to be important cells in modulating both the reabsorption of sodium and also the reabsorption of water. And so with sodium, these principal cells are activated by aldosterone of the RAA pathway. And by moving sodium out of the filtrate and into the blood, water then is going to follow. And then we also have the opportunity of moving water out of the filtrate and back into the blood, complements of principal cells that are activated by another hormone called antidiuretic hormone, or vasopressin. So with the release of vasopressin or ADH, we also have the tubular reabsorption of water. So a lot of opportunities for the refinement of water absorption here in the distal convoluted tubule. And then finally, we have the opportunity for reabsorption of calcium ions. And this happens when parathyroid hormone is released by the parathyroid glands. Parathyroid glands increase the levels of blood calcium, and one of the ways that they help to increase that is to reclaim any lost blood calcium from the filtrate into the blood. So we've discussed the processes of filtration and tubular secretion and tubular reabsorption. We can also alter these as well. And so we can voluntarily take compounds known as diuretics. Diuretics are substances which either increase tubular secretion of water or decrease the tubular reabsorption of water. So let's take a look at what that means over here in a very simplistic schema. So by increasing tubular secretion of water, that means we go from moving a little bit of water from the blood into the tubes to a lot more water from the blood into the tubes. By decreasing tubular reabsorption, we would take what would be a larger volume that was resorbed from the filtrate into the blood, and we reduce that into a lower amount. So the net result of this is more water either stays within the filtrate or more water is moved into the filtrate. By having more water in the filtrate, there's more volume of urine. So diuresis is this process of either keeping or expanding the amount of water in the urine. So a diuretic is any substance that contributes to diuresis, and that is going to be a rather simplistic first-line treatment for hypertension or high blood pressure. So with hypertension, we can reduce blood volume through diuresis and therefore help to reduce the amount of pressure inside of arteries. Examples of diuretics in your own life that you may choose to either consume or avoid are caffeine and alcohol. Caffeine contributes to diuresis two ways. One, it slows water reabsorption, and two, it also increases glomerular filtration rate. So by increasing glomerular filtration rate, you're producing more filtrate, which will lead to more water. Alcohol is an interesting diuretic in that alcohol inhibits vasopressin production. By inhibiting vasopressin production, you are reducing the distal convoluted tubule's ability to reabsorb water, so you slow that, and as a result, more water then stays within the distal convoluted tubule and remains as filtrate for the eventual formation of urine. That's why when people drink either caffeine or alcohol, they seem to urinate in greater proportion to the amount of liquids that they are consuming. So we've discussed the nephron, its histological features, and the physiological processes of glomerular filtration, secretion, and reabsorption. We've discussed the parts of the nephron and what substances are either secreted or reabsorbed in those different parts. And we've discussed how diuretics are going to affect urine formation. Thank you so much for your time, and I'll talk to you in the next video.